Heme synthesis can occur in all tissues, but the cells that produce the largest amounts of heme are hepatocytes and nucleated red cells. In this animation, we will review the synthesis and degradation of heme, as well as defects in heme synthesis that manifest as either hepatic or erythropoietic porphyria. Let's zoom in on the liver, and further to a hepatocyte. The first and last steps of heme synthesis occur in the mitochondria, while the steps in between occur in the cytoplasm. Let's zoom in to a mitochondria. In the mitochondrial matrix, the first step of heme synthesis involves the condensation of the amino acid glycine with succinyl-CoA from the citric acid cycle. This reaction is catalyzed by ALA synthase, the key regulated enzyme of the pathway. It uses pyridoxal phosphate as a coenzyme to form delta-amino-levulinic acid, abbreviated here as ALA. Once formed, ALA is translocated out of the mitochondria to the cytoplasm. There, 2 molecules of ALA are condensed by ALA dehydratase to form porphobilinogen. 4 molecules of porphobilinogen are then condensed by porphobilinogen deaminase, or PBG deaminase, labeled here as PBGD. This reaction forms hydroxymethylbilane. Hydroxymethylbilane is converted by uroporphyrinogen 3 synthase to uroporphyrinogen 3. Uroporphyrinogen 3 is converted by the enzyme uroporphyrinogen decarboxylase to coproporphyrinogen 3. Coproporphyrinogen 3 is then translocated into the mitochondria for the remaining steps of heme synthesis. Here, it is converted to protoporphyrinogen 9 and then oxidized to protoporphyrin 9. Finally, the mitochondrial enzyme ferroketolase adds ferrous iron, or Fe2+, to protoporphyrin 9 to produce heme. Now, it is important to recognize that heme can serve as a feedback inhibitor of ALA synthase. Therefore, heme is not synthesized when a sufficient concentration of heme is already present. Shown here is a summary of the complete pathway of heme synthesis just reviewed. Porphyrias are disorders of heme synthesis that are either acquired or inherited and lead to overproduction and accumulation of porphyrins. Acute intermittent porphyria is caused by defective porphobilinogen deaminase. In the mitochondria, as normal, glycine reacts with succinyl-CoA to form delta-amino-levulinic acid. ALA then enters the cytoplasm, where 2 molecules form porphobilinogen. Normally, 4 porphobilinogens condense to form hydroxymethylbilane, but this step does not occur due to a defective PBG deaminase. This results in an accumulation of porphobilinogen and its substrate, delta-amino-levulinic acid. Acute intermittent porphyria is inherited as an autosomal dominant condition. Patients often present with port wine-colored urine, acute abdominal pain, and psychiatric disturbances. Now, the most common porphyria is porphyria cutanea tarda. This condition is due to a deficiency of the enzyme uroporphyrinogen 3 decarboxylase. Again, heme synthesis proceeds normally until a deficiency of uroporphyrinogen 3 decarboxylase prevents the conversion of uroporphyrinogen 3 to coproporphyrinogen 3. This results in accumulation of uroporphyrinogen 3. UPG3 builds up in the liver. And in the skin. Because porphyrins have a conjugated double bond network in their structures, they absorb light. This causes porphyrins to produce free radicals, which, in turn, cause lesions or blisters on the skin. This photosensitivity is the hallmark presentation of porphyria cutanea tarda. Heme catabolism occurs primarily in the spleen. Let's take a closer look at the spleen. In the macrophages of the spleen, old red blood cells are destroyed, and their hemoglobin is degraded. The heme portion is processed to salvage its iron and eliminate its porphyrin, which is oxidized to carbon monoxide and the green pigment biliverdin. This is the only reaction in the body that generates carbon monoxide. To salvage the iron, it is transported from the macrophages via its protein carrier, transferrin, which delivers it to other organs, such as the liver. Back in the macrophages of the spleen, biliverdin is converted to the yellow-orange pigment bilirubin. Bilirubin is then transported to the liver via albumin. Bilirubin is a nonpolar, water-insoluble molecule. Therefore, it requires transportation through the circulation by a protein carrier. In the liver, the enzyme UDP glucuronyltransferase uses two molecules of the substrate UDP glucuronic acid to synthesize conjugated bilirubin, which is sometimes called bilirubin diglucuronide. This process, called conjugation, converts bilirubin to a polar form, making it water-soluble, and thus able to be excreted in the bile. In clinics, unconjugated bilirubin is referred to as indirect bilirubin, whereas conjugated bilirubin is known as direct bilirubin. From the liver, conjugated bilirubin is secreted into bile, which is stored in the gallbladder. After a meal, bile is then secreted from the gallbladder into the duodenum. In the lumen of the intestine, bacterial flora metabolizes conjugated bilirubin into stercobilinogen and urobilinogen. Stercobilinogen travels through the intestinal network, where it is oxidized to stercobilin and eventually eliminated in the stool. Stercobilin gives stool its characteristic brown color. Meanwhile, urobilinogen is reabsorbed from the intestinal lumen into the circulation and travels to the kidney, where it is excreted as urobilin. This gives urine its characteristic yellow color.